The Nobel Prizes have been awarded this week. It's that time of year again. The Nobel Prizes have been awarded. Hello and welcome back to Science Wrap, the show where we feed you the tastiest new research from the world of science, all wrapped up in an easy to digest package. My name's Miriam, PhD and science lover. This week, the Nobel Prizes have been awarded again. And that actually means something else. It means that it's the 1-year anniversary of Science Wrap. The prize in chemistry this year went to 3 gentlemen: Jean-Pierre Sauvage, Sir Fraser Stoddart, and Bernard Faringer. They have designed and made tiny molecular machines. The first step, in 1983, was Jean-Pierre Sauvage was able to delicately link 2 molecules together in a chain-type manner called a catenane, through a process called mechanical bonding. The second development was in 1991, when Fraser Stoddart took one of these rings and put it onto an axle, which allowed the ring to move. This structure is called a rotaxane. In both of these processes, the 2 molecules are linked somehow, but not strongly so, and that's key. Each molecule can still move independently of one another, and that's what makes them be able to be used as machines. The third step, which is where Bernard Faringer comes in, is making a type of molecular motor where you can put energy into molecules and they spin continuously. Put all these 3 components together and you have a machine that's capable of simulating human muscle, being able to create the world's tiniest car, and in the future being able to develop new specialized materials, new ways of storing energy, and potentially also ways of delivering drugs within the human body. In the field of physiology or medicine, Yoshinori Usumi took out the prize. He discovered and described the mechanism known as autophagy. The word autophagy literally means to eat oneself. But in the field of biology, it refers to a process where a cell can destroy or recycle certain components of itself. A cell might want to do this to get rid of material that's dangerous or damaged. Dysfunction in autophagy mechanisms can lead to some really serious diseases, including cancer. In the 1950s, it was discovered that tiny cells are made up of even tinier subparts, which people called organelles. Now, these organelles help the cell carry out its particular functions, like replication or making energy for itself. The organelles of interest for autophagy, there's 3 of them. So the first is the lysosome, which makes the compounds that can break down things in the cells, like proteins, carbohydrates, or fats. The second is the vacuole, which is kind of like an empty stomach waiting to be filled with stuff to be digested. And the third is called an autophagosomes, and this is like a delivery material to bring the stuff to be digested into the vacuole. Usumi conducted some really clever experiments where he altered the genetic code of yeast cells so that the lysosomes no longer produce the chemicals that break down stuff. This meant instead of the vacuole appearing empty or having stuff be digested in there, it ended up getting filled with those autophagosomes. But by tapping into the genetics of the yeast cell, he also discovered which genes were responsible for regulating the process of autophagy. It was basically like a 2-for-1 deal. He then later went on to characterize these genes in great detail. Last up in the sciences, the prize for physics went to 3 gentlemen: David Thouless, Duncan Haldane, and Michael Kosterlitz. They've used advanced mathematical modeling to explain some really weird states of matter. So you know about the states of matter like solid, liquid, or gas. But there are these other states that have interesting properties, like plasma, hint hint, or quantum condensate. Essentially, states mean how close molecules are to one another and how fast they're moving, so how much kinetic energy those molecules have. In the 1970s, Duncan Haldane and Michael Kosterlitz described that going between states by applying heat isn't as simple as, say, going from solid to liquid. That change in state, or that transition, looks like tiny little vortices. You can kind of imagine it like ships on the water. At low temperatures or calm seas, 2 ships can travel side by side with being quite close to each other. At high temperatures, however, or in turbulent seas, those ships start spinning off into different directions. This whole process is known as the KT transition. In 1983, David Thowlis found that really strange things start to happen when you cool materials down to near absolute zero. We're talking close to minus 273 degrees Celsius. He described what's called the quantum Hall effect, where the ability of a supercooled material to conduct electricity changes in a stepwise rather than a continuous pattern. This all has to do with the idea of topology, which means the integrity of a shape when you twist it or stretch it. You can think about it kind of like a lump of clay. It's still a lump of clay no matter how you move or bend it or what you shape it into. Under particular conditions, this lump of clay starts to get more and more holes in it, going from a ball of clay, so no lumps, to a donut, 1, a pair of glasses, 2, to a pretzel, 3. And it continues like that in a stepwise way. The same is essentially happening to that material. That's all pretty complicated stuff. But the application of it is that we can make supercomputers from these so-called topological materials. Well, that's a wrap on the Nobel Prizes for 2016. More information can be found on the Nobel Prize website. I've linked all the details below. As always, thank you so much for watching Science Wrap, and we'll see you guys next time. Bye!